मोहित सर।
जी सर, बस 1 मिनट और वेट कीजिए, फिर उसके बाद। अभी सर ज्वाइन कर चुके हैं। विवेक सर।
Good morning to Dr. Navneet Verma. Navneet sir. Navneet sir, am I audible?
Sir, good morning.
Good morning, sir. How are you, sir?
Fine, sir. Fine.
गुड मॉर्निंग नवनीत सर।
Good morning. Good morning. साहू कैसे हैं आप?
बढ़िया सर, फाइन सर। वेरी लॉन्ग टाइम यू आर हियर। कैसे हैं सर आप?
बढ़िया बढ़िया, आप सुनाइए।
बस बढ़िया सर, बहुत लंबे टाइम बाद आपको देख रहा हूँ।
गुड मॉर्निंग सर। गुड मॉर्निंग।
Good morning.
नमस्कार सर। विवेक से स्टार्ट कीजिए।
Start कर देना सर।
जी स्टार्ट।
So a very warm good morning to all the honorable guests, our faculty members, and all the students who are with us on YouTube. So first of all,
लाइव टेलीकास्ट कर दिए सर?
live telecast is on, sir.
ऑन, ठीक।
So firstly, I should come up with a brief introduction of Professor Navneet Verma. Professor Navneet is a registered pharmacist and has obtained his B.Pharm degree from Uttar Pradesh Technical University, Lucknow, and did his M.Pharm in pharmaceutics from Bundelkhand University, Jhansi. Over the last 16 years, he is engaged in teaching of pharmaceutics to the undergraduate and postgraduate students. Professor Verma has received number of awards and recognitions from various societies. He was assigned as a completed number of administrative responsibilities of the university successfully. He has obtained his PhD degree in pharmaceutical sciences. 22 M.Pharm dissertations were supervised by him, and 4 PhD degrees has been awarded in his supervisions. 6 research scholars are enrolled for PhD degree reviewer board of his various national and international journals. He has his credit 40 research paper in journals of national and international repute, and 12 papers has so far has been presented in various conferences, seminars, and workshops. He has also organized a number of seminars, conferences, and academic events in university successfully, and is an active member of various professional bodies in India. He is also acting as an additional controller of examination of 10 university. And Professor Verma has around 10 published patents. So with this short introduction, I once again welcome Professor Verma to this faculty development program, and this was the last day. And for our next speaker, may I request to Dr. Divya to please come up with a brief introduction of our honorable speaker.
Okay, sir. Good morning to everyone. So on behalf of IIMT University, I, Dr. Divya Pathak, convey my regards to the eminent speaker of the day. So thank you so much, sir, for accepting our invitation and coming over the FDP. So this is the short introduction to Dr. Sahu. So Dr. Jagdish Kumar Sahu did his B.Pharm from the Jivaji University, Gwalior in 2005. M.Pharm from Department of Pharmaceutical Sciences, Dr. H.R. Gott, Central University, Sagar, MP in 2008. With first class and PhD from the Birla Institute of Technology, Mishra, Ranchi in 2016. So he is having 14 years of the teaching and 7 years of the research experience with an administrative experience of 3 years, superintendent of the examination in IPS College of Pharmacy, Gwalior also. So Dr. Sahu is a recipient of AICTE scholarship for the PG program. For the PG program in 2005. He has received research grant of the 3.58 lakhs from the MPCST Bhopal and 1 lakh from the NMIMS Mumbai. He has published impact factor more than more than 35 research papers and review articles in various journals of highly reputed with cumulative impact factors like 32.41 in SCI Index Journal. Dr. Sahu has 1 Indian patent and 1 Australian patent, and he is recipient of the Best Paper Award 2 times. He has received a tag of Bantam Brand Ambassador for the 2019-20. He is an external examiner and paper setter of the many reputed institute. And he is serving as a nominee of the CPCSA, Government of India since 2017 onwards. He is an active and life member of many professional bodies like APTI, IPR, SPER, etc. His area of interest is synthesis and biological evaluation of new chemical compounds with a special reference to heterocyclic moieties, drug design, analytical methods, developments, and validation. He is currently serving as an assistant professor in the Department of Pharmaceutical Chemistry, School of Pharmacy, Technology, Management, SBKM, NMIMS, Deemed University, Sirpur, Maharashtra. So in addition to academic responsibility, Dr. Sahu is also working as a managing editor, current research in pharmaceutical sciences and international journal published by the EPSA India. He is also reviewer and editorial board member of many reputed journals like Medicinal Chemistry, Research, Journal of Advanced Pharmaceutical Technology and Research, etc. So welcome to sir. Dr. Sahu, over to you.
Okay. Am I audible?
Yes, sir.
Okay. Thank you, madam. Thank you, Dr. Divya, madam, for this wonderful introduction. And I am thankful to organizer of this 7 days EFTP program and entire team of IIMT University Meerut for inviting me here as a guest speaker in today's EFDP. Thank you, ma'am. Thank you, sir. And now it's the time to discuss our topic, what I am going to have shared, repurposing novel therapeutic candidate drugs for combating COVID-19 disease. I hope this PPT is visible to you, ma'am, sir.
It's not visible.
Not right, sir.
Okay, just. Now is it visible, sir?
No, sir.
No, sir.
I think it has to be shared again.
Just a minute, sir.
I'm going to share again. Just a minute. I hope now it is visible.
Yes, sir.
Yes, sir.
Now it's visible.
So now is it visible, ma'am?
Visible,
Yes,
sir.
visible, visible.
Okay, okay. So the topic of my today's presentation is repurposing novel therapeutic candidate drugs for combating COVID-19 disease. So first of all, we have to discuss that how we can repurpose and what is basically repurposing. What is the drug repurposing? So I have changed the slide. I hope change slide is also visible to you. So
Yes, sir.
What is basically drug repurposing, we can say. It can be defined as a process of identification of a new pharmacological indications from old or existing or failed investigational, already marketed, FDA approved drugs or products, and the applications of newly developed drugs to the treatment of diseases other than the drug's original use. Means drugs has been discovered for another use, but now it has been used for some other treatment option. So it involves establishing new therapeutic uses for already known drugs, including approved or discontinued and experimental drugs. So there are various synonyms, we can say, for the drug repurposing, like drug repositioning, we can say, drug retasking, drug reprofiling, drug rescuing, drug recycling, drug redirection, and therapeutic switching. So these are the various synonyms we can say for the drug repurposing. So the drug repurposing means already it has been established for another treatment, but now still it is using for current another disease. So that is repurposing. Again, we are using here. So it has been observed that approximate 30% of the US FDA approved drugs and biologics or vaccines are basically repositioned drugs. Some most successful and best known drugs that have been emerged out the TR approach are sildenafil, minoxidil, aspirin, valproic acid. So just I am giving an example of sildenafil, popularly known as Viagra. So it is basically a phosphodiesterase inhibitor, initially developed for coronary artery disease, angina. It has been discovered for and developed for angina, but now Pfizer has been repurposed this drug for the treatment of erectile dysfunction. So now it is popularly known as Viagra for especially erectile dysfunction. However, it has been developed for the treatment of angina. Another example, metformin. Metformin, it is now popularly known anti-diabetic drug, and it is now widely used in type 2 diabetes mellitus. But initially it has been developed as a cancer therapeutic agent, and however it is still under the process of phase 2 and phase 3 clinical trials. So these are the examples of very common examples of drug repurposing, sildenafil and minoxidil. So next slide, what is the need of drug repurposing? First of all, question arises why it is needed. Actually, the conventional way of the drug discovery, that is drug development. So it is basically more time consuming and very expensive process. So recent estimates suggest that on an average, it takes approximate 10 years and at least $1 billion to bring a drug to market. And moreover, investment in drug development has been gradually increasing as reported by Pharmaceutical Research and Manufacturers of America. So pharmaceutical companies have become increasingly interested in finding new uses for already existing drugs. So that is the finding new use of already known drugs or existing drugs. So this is the process we can refer as drug repurposing or drug repositioning. Okay, next is, so how we can compare with this drug repurposing as drug discovery. So traditional drug discovery versus drug repurposing. So the traditional approach to drug discovery, it involves de novo identification of new chemical entities, which includes 5 stages: discovery and preclinical, safety review, clinical research, FDA review, and FDA post-market safety monitoring. So it is a time-consuming and costly, very costly process, even with high risk of failure also. So on the other hand, there are only 4 stages in drug repositioning or repurposing, which includes compound identification, compound acquisition, development, and FDA post-marketed safety monitoring. So this is an overview that what are the stages of drug development and what are the stages of drug repositioning or repurposing. So you can clearly see that drug repurposing, it consists of only 4 steps, while drug development, it consists of more than 4 stages, 5 stages, and even it is time taking and time consuming process. So what are the advantages of drug repurposing over traditional drug discovery? Why it has become so much important nowadays? So drug repurposing, also known as using of old drugs for new uses. Already drugs are well known to us, and it is currently using for somewhat new purposes. Okay, so it is an effective strategy for finding new indications for existing drugs and is highly efficient, low cost, and riskless. So traditional drug development strategy, it usually includes 5 stages, as already I have discussed. But now in drug repurposing, you can see there are only 4 stages there: compound identification, compound acquisition, development, and finally FDA post-market safety monitoring. So there are only 4 steps, and still it contains 1 to 2 years over the when compared to 8 to 10 years. So it is already, it is not time consuming and very advantageous for us. So due to the fast growth of bioinformatics, knowledge, and biology big data, drug repositioning decreases the time cost of the drug development process significantly. So researchers only need 1 to 2 years to identify new drug targets and 8 years to develop a repositioned drug on an average. So since drugs are already known and drugs are still, we know their mode of action, pharmacology, therapeutic indications, everything. So normally it needs only 1 to 2 years to identify new drug targets for the already known drugs. So now comes on the strategies, that what strategies generally we follow for drug repurposing. So there are 2 different strategies we follow. One is on target, second one is off target. So on target, it means finding new uses of a drug. Already we have known targets, known target we have. So it includes literature, experiments, and positive feature is that it is likely to be compatible with dosing of original drug. So the big exception of privileged compartments is there, ocular and brain. While off target, we are finding new uses of the drug acting through a novel or unanticipated target. It includes some computational work like docking, fingerprint methods, and drugs was not originally optimized for that target. So it need to be very critical of dosing. So this is just an overview that how we can compare on target and off target of drug repurposing. So drug which act on known target, if we know it's therapeutic, non-therapeutic indication. So new therapeutic indication could be done and drug is repurposed. While going to off target, still we know known targets are already there, but we could identify some new targets where it can work. And on the basis of that, we can go to non-therapeutic indication, and on the basis of new targets, we can identify some new therapeutic indications, and similarly in this concern, drug is repurposed. So there are two different ways, on target and off target uses. So on the basis of that, drug repurposing could be done. I hope the point is clear. So for example, just for explaining on target and off target strategy of the drug repurposing, there are two examples. Minoxidil, this is anti-diabetic drug. It is on target profile. So drug acts on the same target and it produces two different therapeutic effects. It is working as antihypertensive and even hair loss drug, and as an antihypertensive vasodilator, it has the property of widening blood vessels and opening potassium channels also. So it allows more oxygen, blood, and nutrients to the hair follicles. And this pharmacological action helps it use in the treatment of male pattern baldness, which is which is very common in men. Androgenic alopecia, we can say for that. So minoxidil is a common example of on-target kind of strategy. While going to off-target strategy, aspirin is a very good example. Aspirin generally used as in the non-steroidal anti-inflammatory drug in the treatment of various pain and inflammatory disorders. So it also suppresses blood coagulation, clot formation by inhibiting the normal functioning of the platelets, means it is anti-platelet drug. So it is therefore used in the treatment of heart attacks and strokes also. So another new use of aspirin in the treatment of prostate cancer has also been reported. So here you can see it is there are some different ways it is using. So it is a very good example of off-target strategy of drug repurposing. So come to the approaches. What are the approaches of drug repurposing? So there are basically two different kind of approaches are there. One is activity-based approach and in silico based approach. So activity-based approach, experimental in vitro and in vivo screening. target based and means experimental screening would be there when we can go to activity based approach, while in silico based approach it is totally involve computational data, computational screening it involves. So this is in silico based approach. Activity based approach, in that target based and cell based or organism based screening assay would be done, while in silico we generally follow protein target based screening. So in activity based approach, generally we do not require any structural information of target protein and drug induced cell or disease phenotypic information. But in silico based, we need structural data of the target proteins and even drug induced cell oblique disease phenotypic information. We highly need for that. So activity based approach is somewhat time and labor consuming, while in silico we have sufficient time. It is means time and labor efficient approaches there. Lower rate of false positive hits during the screening, that is activity based approach. It follow very low rate of false positive hits during the screening, while in silico based approach it has higher rate of false positive hits during the screening. So rate of failure is higher in this kind of approach, in silico based approach. So this is just an overview that how we can go to the different approaches, while one is experimental screening and another one is in silico screening. So experimental screening, when we go to, there are biological targets and cell organism based. So generally we use in vitro assay and in vivo screening for using these approaches, while in silico it is totally computational data. So we use pharmacophore modeling and molecular docking studies for the for the development of this kind of strategy. So as you know that molecular docking data, generally we only predict 60%. For 60% prediction is that it is it may be by using this study. So generally while going to in silico screening, we generally include experimental data there. So in silico screening and this is experimental screening, both strategies are there. So come to the methodologies of drug repurposing. So basically, three different type of methodology we can use for drug repurposing. One is drug oriented, target oriented, and disease or therapy oriented. So in the drug oriented methodology, the structural characteristic of drug molecule, that is biological activities, adverse effects, and toxicities are evaluated. This strategy is meant for identifying molecules with biological effects based on cell or animal assays. So this type of repositioning methodology is based on traditional pharmacology and drug discovery principles, where studies are usually conducted to determine the biological efficacy of drug molecules without really knowing about the biological targets. Come to the next methodology, that is target-based methodology. It comprises in silico screening or virtual throughput screening of drugs or compounds from drug libraries, databases such as ligand-based or molecular docking, followed by in vitro and in vivo high throughput screening of the drugs. So there is another approach, that is disease or therapy oriented methodology in drug repurposing, when there is more information on the disease model is available. So this type of drug repurposing is only applied when more information on the disease model is available. And in this case, drug repurposing can be guided by the disease and or treatment based upon availability of information given by proteomics, genomics, metabolomics, and phenotypic data. So it therefore requires construction of specific disease networks, recognizing genetic expression, considering key targets, identifying disease causing protein molecules related to cell and metabolic pathways of interest in the disease model. So there are three different methodologies we have followed for the drug repurposing. Come to the therapeutic targets. What are the therapeutic options for COVID-19 treatment? So as of last year, October 2020, as on the data, there have been 40 million cases of COVID-19 and over 10 lakh deaths worldwide. So it is very useful to discuss about the current therapeutic options for the COVID-19 treatment. So there are different categories by which we can go through that. There are some antiviral drugs, antimalarial drugs, antidiabetics, glucocorticoids, anti-parasitics, anti-tuberculosis drugs, anti-asthmatic, anti-thrombotic, and different category of drugs. Even anti-cancer agents are also there. So they different treatment options under different categories. So we can go through what are the current treatment options. So drugs for the COVID-19. So there are three different categories. First of all, discontinued drug therapies, current approved drug therapy, and proposed drug therapies. So still, some discontinued drug therapies like hydroxychloroquine. It is the first drug we have used for some cases in the treatment of COVID-19. And chloroquine, lopinavir, ritonavir. So these are the earlier therapies which have been used for the treatment, but now it has been discontinued. Current approved drug therapy, that is remdesivir. It is for COVID-19, and there are some other agents are available which are not right now not approved, but still the work going on, that it is under the process of approval. So third one is proposed drug therapies, means what the proposals are still under consideration. So there are some categories like antivirals, diadenosine, favipiravir, umifenovir, sofosbuvir, oseltavir. So these are the antivirals. Corticosteroids are there, dexamethasone, hydrocortisone, methylprednisolone. Some antiparasites are there like ivermectin, nitazoxanide, niclosamide, diethyl carbamazine, and even some immunotherapeutic drugs like tocilizumab, infliximab, bevacizumab, dupilumab, human monoclonal antibodies, that is 4T7D11, and type 1 interferons are there, and some other drugs which are still under consideration, like chemostat, metformin, gliptins, rifampicin, and some herbal drugs are still there, which current working is going on. These are the proposed drug therapies only. So now we can discuss one by one. Sir, I hope the slides are properly visible and even slide change is visible to you.
Visible, sir, visible. It's okay.
Okay, sir. So come to the very commonly known drug favipiravir. It comes under antiviral category. So it is extensively investigated for the treatment of several infections of Nipah, Ebola. So as favipiravir is originally developed for the treatment of Nipah, Ebola, but still it has been using and even used for COVID-19 treatment. So this is another example of drug repurposing, and so it has been developed for Ebola, Lassa, and influenza viruses. But now in Feb 2020, it was being studied in China for experimental treatment of emergent COVID-19. So trials also being going on and even planned in Japan. So a study on 80 people in comparison of lopinavir or ritonavir, it has been found that reduced viral clearance time, and that 91% of people have improved CT scans with fewer side effects. So the drug has been approved for use in clinical trials of COVID-19 disease in China. There are some other drugs, as you know, remdesivir, currently approved drug that is significant broad spectrum activity for against coronavirus, Ebola, and even hepatitis B. There are lopinavir and ritonavir are there. It is a fixed dose combination that is used for first line treatment of HIV or AIDS. There are some another drugs, chloroquine and hydroxychloroquine, under the anti-malarial category. So these are basically quinolone derivatives conventionally used as anti-malarials, anti-amoebics, and anti-arthritics. So till 14th April 2020, US FDA granted emergency use authorization to 25 drugs for COVID-19 treatment, and 10 drugs were selected for cities, whereas the off-label use of hydroxychloroquine and off-label use of hydroxychloroquine facilitated early treatment for COVID-19 patients. There are some anti-parasitics also, like ivermectin, niclosamide, and diethyl carbamazine. There are some antibiotics like picoplanin, azithromycin, and amphotericin B. Amphotericin B, it is a very, in the liposomal form, it is very effectively used for the mucormycosis. Fungal infection normally happened after, normally caused after the COVID-19. And there are some immunotherapeutic drugs like tocilizumab. It is a human monoclonal antibody indicated for autoimmune disease like rheumatoid arthritis, prostate cancer, whatever. It is, it was repurposed for COVID-19 due to its activity on the virus. So this is serilumab that is used for rheumatoid arthritis in adults. So it was repurposed for COVID-19. So there are some miscellaneous agents like metformin. It is an oral drug indicated for treatment of type 2 diabetes and polycystic ovary syndrome. There are some nafamostate serine protease inhibitor. There are some Indian herbs, just like ashwagandha, mulethi, and some other clinical tested for activity against SARS-CoV-2. Even Ayush laid down measures to boost immunity against SARS-CoV-2 using Ayurvedic ingredients like black pepper, cinnamon, clove, cumin, coriander, dry ginger, garlic, turmeric, and tulsi. And there are some homeopathic drugs like arsenicum album, calcirola, and some nitrium nux vomica, silica, hypersulfur, and phosphorus were indicated for COVID-19 treatment. So these are some miscellaneous agents which have been repurposed for the COVID-19 users. Even there is vitamin D. It is as an adjuvant in COVID-19 treatment due to its supportive action in the treatment of ARDS and systematic inflammatory response disease, syndrome, HIV infection, and influenza. So there are amethine is an ipecac alkaloid. It is also proposed for COVID-19 treatment due to its antiviral effects against SARS and MERS coronavirus. So still you can see there are some herbal drugs also are useful for the this COVID-19 diseases. There is combination therapies also there. So in combination therapy, we can go zinc supplementation complements for means chloroquine and hydroxychloroquine treatment in elderly patients and patients with comorbidities such as diabetes, cardiovascular and pulmonary diseases, zinc, etc. And on DCGI approval, Glenmark Pharmaceuticals started even phase CT trial of favipiravir and umifenovir combination on 158 COVID-19 patients in India also. Cimetidine and famotidine, conventionally prescribed for GERD, displayed anti-inflammatory and lung protective action, whereas fenofibrate and benzafibrate minimized viral load and increased lung protective actions. So combination therapy is also useful for repurposing COVID-19 diseases. It's all from my side. Thank you so much for inviting here as a speaker on this platform. And now, if any questions and queries, if you have, you are welcome to ask.
Thank you, sir. Thank you so much, sir.
Thank you, sir. Thank you so much. Sao, sir, there is one query.
Yes, sir.
The person seems to be very normal, sir. He is not having fever. He is not having cold. He is not sneezing. But if he has gone through RT-PCR report, the report is positive. Okay. So it may be a kind of a symptom, sir, or it may be there was something wrong with the report or in analysis.
As per the guideline, actually, sir, we have to follow in this case guideline. So if even there are no symptoms like no coughing, no sneezing, whatever, so at least we have to follow the guidelines. We have to make we have to consult the physician also, and we have to become quarantine for that. Even because even report is saying positive, so still we have to follow the guidelines for that.
If a person is having a RT-PCR report positive,
RT-PCR report is
but the person is not having any symptoms, so better option that the patient has to be isolated.
Yes, sir.
He
Actually,
includes under observation.
yes, yes, sir. Even as per the recent guideline of government, he is saying that after 7 days, even after 3 days, if there is no fever, then you can assume that you are COVID negative. Yeah. In continuation 3 days, there is no fever or no symptoms are there, so you can assume that you are negative, and you have to perform again. Even there is no need to go again for the test also, as per the recent guideline.
So
तो
this
आपकी एक्सक्यूज मी विवेक सर, ये तो गवर्नमेंट की गाइडेंस है सर, आपकी क्या गाइडेंस है? एज पर हम
Sir,
तो
what
आपका करना चाहते हैं, ये तो गवर्नमेंट की गाइडेंस है ये तो।
yes, sir, what I think that safe side, if I think report is saying positive, no doubt, but being a safe side, I think at least for 7 days I have to become isolated from others. That is our guideline, and just follow the just you can consult the physician also, safe side. Just take more fluids, vitamin C, vitamin D, whatever. Even
sir, how to distinguish between a viral fever and a COVID-19, sir?
yes, sir. Actually, the most symptoms are same, no doubt, because both are viral infection. COVID-19 is also a virus, and common cold is also a virus. But what you can see that if even I have suffered 2 times from viral infection, and I have means gone for the test also. However, it has been negative, but still I have consulted with the physician and uh, uh, treated according as per their advice also. So I think safe side, we have to make the consultation with the physician and just being isolated for some time period, what he suggests.
Yes, sir.
Okay, thank you so much, sir. Thank you so much. Now, on the behalf of Faculty of Pharmacy, IIMT University Meerut, a very warm good afternoon to everyone. Vivek sir, I am audible? Clear?
Yes, sir.
Okay, thank you, sir. Thank you so much. A very warm good afternoon to everyone. I would like to my most sincere thanks to the Honorable Chancellor Sir, IIMT University Meerut, Sri Yogesh Mohan Ji Gupta Sir, and Honorable Vice Chancellor Ma'am, Professor Deepa Sharma Ma'am, and Honorable Pro Vice Chancellor Sir, Professor Satkumar Bansal Sir, Honorable Registrar Sir, Professor BP Rakesh Sir, and I extend a very hearty vote of thanks to respected and most distinguished our Chief Guest, Secretary, Central Committee of Association of Pharmaceutical Teachers of India, APTI, Professor Ramakdang Sir, and guest of honor, Professor Navneet Verma Sir, Dean, Pharmacy Academy, IFTM University, Muradabad, and our all guest speaker who is spared his valuable time from their busy schedule to grace the e-faculty development program on emerging trends and challenging in pharmaceutical sciences and healthcare. Guest speakers, thank you so much, Dr. Arun Kumar Mishra sir, Pharmacy Academy, IFTM University, Muradabad, Dr. Kamla Pathak Madam, Faculty of Pharmacy, Uttar Pradesh University of Medical Science, SAFI, and Dr. Satprakash Singh, Institute of Pharmacy, Dr. Ram Manohar Lohia, Awadh University, Ayodhya, and Dr. Amrita Mishra ma'am, School of Pharmaceutical Sciences, IFTM University, Muradabad, and Dr. Gaurav Tiwari sir, Kirvir Singh, Institute of Technology, Kanpur, and Dr. Jagdish Kumar Sahu sir, NIMS University, Maharashtra, and Dr. Ruchi Tiwari ma'am, PSIT, Kanpur. And I would like to express our deep gratitude to the all respected Dean and HOD of various departments for his or her their presence in this FDP. And I would also thank Dr. Amit Kumar sir and School of Pharmaceutical Sciences, IIMT University, and Professor Sachin Kumar sir, IIMT College of the Medical Science, of their guidance and moral support. I am very happy to express a vote of thanks to all respected and dear faculty members, College of Medical Sciences and School of Pharmaceutical Sciences, who has made this event a grand success through their motivation and dedication. Thank you, Vivek sir. And my hearty thanks to the all participants, faculty member of different colleges and university, and all the members of organizing committee, because of whom we made this online FDP possible. And we will send e-certificate to all registered participants of this APTI sponsor FDP as soon as possible. And last but not least, a great honor to propose the vote of thanks to all who have helped us in making this event a very resounding success. Once again, thanks a lot to everyone. Namaskar. Jai Hind.
Sahu sir,
Yes, sir.
just make me a host, sir.
Okay, okay, sir.
Thank you, Vivek sir. Thank you. Special thanks.
Thank you, sir. Thank you, Mohit sir. Once
Atul
again.
sir.
Sir, it is having options like, okay, make host. Yes. Okay.
Now I am a host, sir.
Yes.
Once again, thank you to all the participants who have participated once again, and once again, a very thanks to all the students of IIMT University and various other universities who are with us on YouTube Live. Thank you, sir. Thank you.
Thank you, sir.